Today we're discussing Chapter 22, covering the lymphatic system. So the ability to ward off pathogens, anything that causes a disease, is going to be called resistance. So the lack of resistance, an inability to ward off disease, is called susceptibility. And resistance to a disease can be grouped into two broad areas. We've got non-specific resistance. This is also called innate immunity. And we've got immunity, which is also called adaptive immunity. So in innate immunity or non-specific resistance, this is going to be present at birth, and it's going to include defense mechanisms that are going to provide general protection against invasion by a wide range of pathogens. On the other hand, immunity or adaptive immunity is going to involve the activation of specific lymphocytes that are going to be involved in combating particular pathogens or other foreign substances. So the body system that carries out this immune response is called the lymphatic system. And the lymphatic system is going to consist of a fluid that's called lymph, and this is going to flow within the lymphatic vessels. And there are going to be several structures and organs that are going to contain lymphatic tissues. These are going to be specialized reticular tissues that are going to contain large amounts of lymphocytes. These are also going to be, this lymphatic tissues can also be bone marrow, which is also an excellent site for lymphocyte production. So interstitial fluid and lymph are essentially the same. The only really big difference or way that we distinguish between the two is based on location. So lymph is going to be within the lymphatic vessels. That's really the only difference between lymph and interstitial fluid. So there are three main functions for the lymphatic system. The first is going to be to drain any sort of excess interstitial fluid. The second is to transport dietary lipids from the GI tract into the blood. And then the third is going to be to protect against invasion through immune responses. The lymphatic vessels are going to begin as lymphatic capillaries, and these are typically going to be closed at one end. You can see that sort of here in our little animated picture. Lymphatic capillaries are going to be located between the cells of many different types of tissue. And these lymphatic capillaries are going to merge to form lymphatic vessels. And these are going to have thin walls and many vessels. So at intervals along the lymphatic vessels, lymph is going to flow through lymph nodes, and these are going to be encapsulated bean-shaped organs that are going to consist of masses of B cells and T cells, those immune cells. Lymphatic capillaries are going to be found throughout the body, except for and particularly avascular tissue. This is going to be things like the central nervous system, as well as portions of the spleen and the red bone marrow. Lymphatic capillaries are also going to have a slightly larger diameter than what you would see in blood vessels or in blood capillaries. And they are going to have an overlapping endothelial cells. And these are going to work as a one-way valve for fluid to enter the lymphatic capillaries. And then anchoring filaments are also going to attach endothelial cells to the surrounding tissue. So here you can see our little anchoring filaments that are going to attach our lymphatic capillaries to that surrounding tissue. So a lymphatic capillary is going to be the villus of the small intestine is going to be called a lacteal. And it's going to function in transporting digested fats from the small intestine into the blood. That brings us into the lymphatic trunk as well as the associated ducts. So from those lymphatic vessels, the lymph is going to pass through lymph nodes and then into the lymphatic or the lymph trunks. And these lymph trunks are going to include the lumbar, the intestinal, the bronchiomediastinal, as well as the subclavian and jugular trunks. Lymph trunks are then going to merge to form either a thoracic duct or the right lymphatic duct. So those are our two options there. The thoracic duct is going to begin as a dilation called the cisterna chyli, and this is going to be the main collecting duct of the lymphatic system. The thoracic duct is going to receive lymph from the left side of the head, as well as the neck, the chest, and the upper portion of the extremities. It's also going to receive lymph from the entire body below the ribs. So this thoracic duct is going to make up a large portion of the area that's going to be drained by the thoracic duct. So it's a lot of it. And then the right lymphatic duct is going to take up a much smaller region. You can see that here in this little image. The right lymphatic duct is going to drain lymph from the upper right side of the body, and it drains lymph into the venous blood via the right subclavian vein. So now moving forward, let's look at the formation and flow of lymph. So the interstitial fluid is going to drain into the lymph capillaries. We've established this. The passage of lymph is from the arteries and blood capillaries in the blood to the interstitial space in the interstitial fluid, to the lymphatic capillaries where it becomes lymph, to the lymphatic vessels, to the lymph trunk, to the thoracic duct, or to the right lymphatic duct. And then from there, they're going to go into the subclavian veins, back into the blood. So you can see this kind of flow of lymph laid out a little bit more simply up at the top here, which is nice and simple. So the same two pumps that aid in the return of venous blood to the heart are going to also maintain the flow of lymph. Remember, we've got the respiratory and the skeletal muscle pump. And lymph is going to flow as a result of this milking action of the skeletal muscle contractions, as well as the respiratory movements. It's also going to be aided by lymphatic vessel valves that are also going to help to prevent backflow of that lymph. So we're going to start looking at the primary lymphatic organs and the tissues that are all going to make up the lymphatic system. The primary lymphatic organs are going to be the site where stem cells are going to divide and become immunocompetent. That means that they are going to be able to mount an immune response. And the primary lymphatic organs are going to be the red bone marrow and the thymus. Pluripotent stem cells that are found in the red bone marrow are going to give rise to mature immunocompetent B cells and to pre-T cells. Those pre-T cells are then going to migrate to the thymus, where they're going to develop further and become an immunocompetent T cell. The secondary lymphatic organs and tissues are going to be the site where most immune responses are going to occur. These are going to include the lymph nodes, the spleen, and the lymphatic nodules, also called lymphatic follicles. And the thymus, the lymph nodes, and the spleen are going to be considered organs because each of them is going to be surrounded by a connective tissue capsule, while the lymphatic nodules are not considered organs because they lack this capsule. So most immune responses are going to occur in these secondary immune tissues. So let's look at these individual lymphatic organs and tissues individually here. Starting with the thymus. All right. So the thymus gland is going to lie between the sternum and the heart, and functionally, it's going to be involved in immunity as the site of T cell maturation. So an enveloping layer of connective tissue is going to hold these two lobes of the thymus closely together, but a connective tissue capsule is going to enclose each of these lobes separately. The extensions of the capsule are called trabeculae, and these are going to penetrate inward and are going to divide each lobe into lobules. Each thymic lobule is going to consist of a deeply staining outer cortex and a lighter staining central medulla. All right. So just based on, we're able to really see the difference here based on how they stain. It's very distinct. We'll look at this in the lab later today. So, starting with the cortex, this is composed of a large number of T cells, as well as some scattered dendritic cells, some epithelial cells, and some macrophages. Those immature T cells, or pre-T cells, are going to migrate from red bone marrow to the cortex of the thymus, and from there they are going to proliferate and begin to mature. The dendritic cells are going to be derived from monocytes, and these are going to assist in the maturation process that these pre-T cells are going to undergo. Each of the specialized epithelial cells, like I said, each of the specialized endothelial cells that are found in the cortex are going to have several long processes that are going to surround and serve a framework for as many as about 50 T cells. These epithelial cells are going to help educate or train up essentially the pre-T cells, and this process is known as positive selection. Additionally, they are going to produce thymic hormones that are going to be involved in the maturation of those T cells. Only about 2% of the developing T cells are going to survive in the cortex, and the remaining cells are going to die via apoptosis. Thymic macrophages are going to be the cells that are responsible for clearing out the debris of all these dead and dying cells. And the surviving T cells, anything that makes it through the maturation process, are then going to enter the medulla of the thymus. So the medulla is going to consist of a widely scattered and more mature T cells, as well as epithelial cells, those dendritic cells, and the macrophages. So some of the epithelial cells are going to become arranged into concentric layers of flat cells that are going to degenerate and are going to become filled with keratohyalin granules, as well as some keratin. And these clusters are going to be called thymic corpuscles or Hassel's corpuscles. So although their role is not entirely certain, we don't really know what they are involved in, we think that it's thought that they might serve as a site of T cell death in the medulla. T cells that then leave the thymus via the blood are going to migrate to lymph nodes, as well as the spleen and other lymphatic tissue. And this is from there they're going to colonize various parts of these organs and tissues. So that's all we have for the thymus. Moving forward then to the secondary lymphatic organs and tissues. This is going to be made up of the lymphatic nodules, the spleen, and the lymph nodes. So starting with lymph nodes, these are going to be kind of similar to the thymus in that they are going to be covered by capsules of dense connective tissue, and these are going to extend into the nodes. The capsular extensions are also called trabeculae, and these are going to divide the nodes into compartments, and it's also going to provide support as well as a route for blood vessels into the interior of the node. And then internal to that capsule is going to be a supporting network of reticular fibers as well as some fibroblasts. The capsules, trabeculae, reticular fibers, and fibroblasts are all going to constitute the stroma. That is, it's the supporting framework of connective tissue that's going to make up this lymph node. Next is the parenchyma, and this is going to be the functional part of the lymph node. And this is divided into the superficial cortex and the deep medulla. The cortex is going to consist of an outer cortex and an inner cortex, and within that outer cortex are going to be egg-shaped aggregates of B cells, and these are called lymphatic nodules or lymphatic follicles. So a lymphatic nodule is going to consist primarily of B cells, and this is going to be called a primary lymphatic nodule. Most lymphatic nodules in the outer cortex are going to be secondary lymphatic nodules, and from this, these are going to form in response to an antigen, as they're also going to be the site of plasma cell and memory B cell formation. So after the B cells in a primary lymphatic nodule recognize an antigen, the primary lymphatic nodule is going to develop into a secondary lymphatic nodule. The center of a secondary lymphatic nodule is going to contain a region of light staining that's called the germinal center. And in the germinal center, there are going to be B cells, as well as follicular dendritic cells and macrophages. So when the follicular cells or follicular dendritic cells present an antigen, the B cells are going to start to proliferate and are going to divide into antibody-producing plasma cells, or they're going to develop into memory B cells. The memory B cells are going to persist after an initial immune response and are going to remember having encountered that specific antigen. So the region of a secondary lymphatic nodule that surrounds the germinal center is going to be composed of dense accumulations of B cells, and these are going to migrate away from their site of origin within that nodule. The inner cortex of our lymph node does not contain any lymphatic nodules. It's going to consist mainly of T cells and dendritic cells that have entered that are going to enter a lymphatic nodule from other tissues. The dendritic cells are going to present antigens to T cells, and this is going to result again in the proliferation of those T cells. The newly formed T cells are then going to migrate from the lymph node to areas of the body where there's going to be an antigenic activity. The medulla of a lymph node is going to contain B cells as well as antibody-producing plasma cells that have migrated out of the cortex of the medulla and the macrophages. The various cells that are embedded in a network of reticular fibers so these various cells that are found in the lymph node are going to be embedded in a network of reticular fibers and reticular cells. Lymph nodes are essentially going to function as a type of filter. That's why they've got all those reticular fibers. So as lymph enters one end of the lymph node, any sort of foreign substances is going to get trapped by those reticular fibers within the sinuses of the node. Then the macrophages are going to come in and destroy any sort of foreign substances via phagocytosis, and then the lymphocytes are also going to be able to destroy other foreign invaders based on or by using immune responses. So this filtered lymph is then going to leave the other end of the lymph node. So in one side, out the other. The slow flow of lymph within the lymph node is going to allow additional time for lymph to be filtered, and all lymph is going to flow through multiple lymph nodes on its path through those lymph vessels. So it's just going through multiple rounds of filtration. And it is quite the slow passage or filtration process of this lymph through the lymph node, so it takes it a good little while. So next up we have the spleen. This is going to be the largest mass of lymphatic tissue that's found in the body, and this is going to be located in the left hypochondriac region between the fundus of the stomach and the diaphragm. So a capsule of dense connective tissue is going to surround the spleen, and it's going to be covered in turn by serous membrane. This is also called the visceral peritoneum. Trabeculae are going to extend inward from the capsule, and the capsule plus the trabeculae, as well as the reticular fibers and fibroblasts, is going to constitute the stroma, or functional part, of the spleen. The parenchyma of the spleen is going to consist of two different types of tissue, one called white pulp and then red pulp. So those are our two types. White pulp is going to be lymphatic tissue. It is going to consist mostly of lymphocytes and macrophages that are going to be arranged around branches of the splenic artery called the central arteries. All right. The red pulp is going to consist of blood-filled venous sinuses, as well as cords of splenic tissue called splenic cords or Billroth's cords. Splenic cords are going to consist of red blood cells, macrophages, lymphocytes, plasma cells, and granulocytes. And veins are going to be closely associated with this red pulp. Here we can see the difference. This is after it's been stained, and it looks very distinct, very different. We've got our red pulp, which is lighter in color, and then our white pulp, much darker staining. And we've got our little central artery right there. So, blood is going to flow into the spleen through the splenic artery, and it's going to enter the central arteries of the white pulp. Within the white pulp, B cells and T cells are going to carry out immune functions that are basically similar to what we would see in a lymph node. And then the spleen macrophages are going to destroy any sort of blood-borne pathogens, again, via phagocytosis. Within the red pulp, the spleen is going to perform three main functions that are related to the blood cells. The first is that it's going to remove blood cells. It's going to be removal of blood cells by macrophages of any sort of ruptured or worn out or old or defective blood cells and platelets. Then we're going to have storage of platelets, where up to 1/3 of the body's platelet supply is going to be stored in the spleen. And then last but not least, we have the production of blood cells, or hemopoiesis, during the fetal life. So when you're cooking inside mom, this is going to be where you're producing most of your blood cells. Next, we have the lymphatic nodules. These are going to be masses of lymphatic tissue that, unlike lymph nodes, are not going to be surrounded by any kind of capsule. Right? They are going to be scattered throughout the lamina propria of the mucous membranes that are lining the gastrointestinal, the urinary, and the reproductive tracts, as well as in the respiratory airways. Lymphatic nodules that are in these areas are also referred to as mucosa-associated lymphatic tissue, also called MALT for brevity's sake. So those are our main organs and tissues that are associated with the lymphatic system. So now, moving forward, we're going to look at the different types of immunity that are provided by this lymphatic system, starting with innate immunity. This is going to refer to a wide variety of bodily responses that are going to serve to protect us against any kind of invasion by a wide variety of pathogens and their sort of associated toxins. So we're born with this kind of immunity. We have it from the very start. We have two major lines of defense. We've got the skin and the mucous membranes. This is our first line of defense, and this is going to act as a barrier to keep out any kind of, hopefully to keep out any sort of pathogens from just entering the tissue. And then we have our internal defense. This is our secondary line of defense. So the skin and the mucous membranes are going to have two main types of defense. We've got our chemical and our mechanical defenses. So for the skin and the mucous membranes, our mechanical defenses are going to be physical barriers. There's going to be things like skin, the mucous membrane, as well as anything that's going to clear stuff out. Things like tears, saliva, mucus, as well as the cilia, the epiglottis, the flow of urine, as well as defecating and vomiting. And then chemical defenses are going to be things like sebum, lysosomes, and the gastric juices that are going to help to sort of kill anything or, repel anything that might be making it in there. Our internal defenses, we're going to have several different types. We've got antimicrobial proteins, phagocytic cells, natural killer cells, inflammation, and fever. Those are our 5 main options we have for our internal defense. So, starting with antimicrobial substances. So lymphocytes and macrophage cells that are infected with viruses are going to produce proteins called interferons, or IFNs for an abbreviation there. So once produced and released from virus-infected cells, our interferons are going to diffuse to uninfected neighboring cells and are going to bind to surface receptors. And this is going to result in those uninfected cells causing them to synthesize antiviral proteins that are going to interfere with or hopefully inhibit that viral replication. So a group of about 20 proteins that are present in blood plasma and our cell membranes are going to comprise the complement system. So when activated, these proteins are going to complement or enhance certain immune, allergic, and inflammatory reactions. So iron-binding proteins are going to remove iron from the bodily fluids, thereby inhibiting any sort of microbial growth. And antimicrobial substances are also going to be peptides that can produce antimicrobial activities, and are able to attract dendritic and mast cells. Next, for our internal defenses, we have the natural killer cells, or the NK cell. These are lymphatic these are lymphocytes that are going to lack the membrane molecules that are going to be necessary to identify them as either a T or a B cell. But they have the ability to kill a wide variety of infectious microbes. That's just where they get their name. So NK cells are sometimes going to release porphyrins, and these are going to insert into the plasma membrane of a microbe, and it's going to make the membrane leaky so that the cytolysis is going to occur. So like breaking open of the cell membrane. It causes that microbe to die, essentially. Other granules of natural killer cells are going to release granzymes, and these are going to be protein digesting enzymes that are going to induce the target cell to undergo apoptosis or self-destruction. So these are granzymes. These are basically enzymes that are going to make the cell hit the self-destruct button. So this type of attack is going to kill infected cells, but not the actual microbes that are within the cell. This the released microbes then may or may not still be intact, but they can be destroyed by phagocytic cells. Speaking of phagocytic cells, these are going to be specialized cells that are going to be involved in performing phagocytosis. And this is basically just the ingestion of microbes or other particles such as cellular debris. The two major types of phagocytes are going to be neutrophils and macrophages. So when an infection occurs, neutrophils and monocytes are going to migrate to the infected area. And then during this migration, the monocytes are going to enlarge and develop into actively phagocytic macrophages. And these are called wandering macrophages. Other macrophages are going to stand guard over specific tissues. They're not going to be moving through the system. So these are called fixed macrophages. So among the fixed macrophages are those histocytes. These are going to be connective tissue macrophages, as well as stellate reticuloendothelial cells, or Kupffer cells, that are found in the liver. We also have alveolar macrophages that are found in the lung, microglial cells that are in the nervous system, as well as tissue macrophages that are found in the spleen, the lymph nodes, as well as in the red bone marrow. So in addition to being an innate defense mechanism, phagocytosis is going to play a really important role in adaptive immunity as well. So let's look at the steps that are involved in phagocytosis, and there are 5 all total. So step 1 is going to be chemotaxis. This is going to be a chemical stimulation that's going to result in the movement of a phagocyte to a damaged site, or a site of damage. So chemicals that are going to attract these phagocytes might come from the invading microbes themselves, or from the white blood cells, already kind of damaged tissue cells, or also possibly from activated complement proteins. So adhesion is our second step, and this is going to occur when attachment of the phagocyte to that microbe or other foreign material occurs. So the binding of our complement proteins to an invading pathogen is going to result in an enhancement of this adherence. Our third step is ingestion. This is when the plasma membrane of the phagocyte is going to extend projections called pseudopods that are going to essentially engulf that microbe in a process that's called ingestion. So when the pseudopods meet, they're going to fuse and surround that microorganism with a little sac called a phagosome. Next, we have digestion. So once that phagosome enters the cytoplasm, it's going to merge with the lysosome and to form a larger structure called a phagolysosome. And the lysosome is going to contribute lysozyme, which is an enzyme that's going to break down those microbial cell walls. It's also going to contribute other digestive enzymes that are going to degrade the carbohydrates, the proteins, the lipids, and the nucleic acids that are associated with that little microbial cell. Phagocytes are also going to form lethal oxidants, such as superoxide anions, as well as hypochlorite anions, and hydrogen peroxide, in a process that's called oxidative burst. And this is also going to help to result in the breakdown, and essentially killing and digestion of that little microbial cell. Our last step in phagocytosis is going to be killing. And this is when the chemical onslaught that's provided by the lysozyme, the digestive enzymes, and that oxidative burst within the phagosome is going to result in essentially the killing of that microbe that's been ingested. So any kind of materials that cannot be degraded any further are going to remain in the phagosome as a structure called a residual body. And they're just going to kind of hang out here for the most part. Our next type of internal defense is inflammation. This is a non-specific defense response of the body to any sort of tissue damage. So among the conditions that may produce inflammation are pathogens, any sort of abrasion, chemical irritations, any sort of distortion or disturbance of cells, as well as extreme temperature. So inflammation is basically an attempt by the body to dispose of microbes or toxins or any sort of foreign materials at the site of injury. And this is done in order to prevent the spread of this injury to other tissues. It's also going to be really important in preparing the site for tissue repair. And this is done in an attempt to restore tissue homeostasis. Tissue homeostasis. There are going to be certain signs and symptoms that are associated with inflammation, and these can be remembered using the acronym PRISH. So you've got P for pain, and this is due to the release of various chemicals that are going to be registering as pain. R is for redness. This is because there's going to be more blood that's rushing to that affected area. I is for immobility. This is going to result from some loss of function in severe inflammation if something is really bad. S is for swelling. This is going to be caused by an accumulation of fluids. And H is for heat. And this is going to be due to that increased blood rushing to that affected area. So that is our inflammation. There are three main steps in inflammation. We're not going to get super in-depth with these, but just know that the inflammatory response is going to have three basic steps. The first is vasodilation and increased permeability of blood vessels. Then we have emigration, where we're going to have the exiting of those phagocytic cells from the blood vessels into that interstitial fluid, where they're going to be involved in kind of tidying up the area so that tissue repair can occur, which is our third step. So two immediate changes are going to occur to blood vessels in a region of tissue injury. All right, we're going to have vasodilation of arterioles, so an increase in blood vessel diameter. We're also going to have increased permeability of the capillaries. This is preparing those capillaries so that emigration can occur for those phagocytic cells. Vasodilation is going to allow for more blood to flow through the damaged area, and it's also going to, that increased permeability is going to allow for defensive proteins, such as antibodies and clotting factors, to enter the injured area from the blood. So an increase in blood flow is also going to help to remove any sort of microbial toxins or dead cells that might be in the area. So there are several different types of substances that can contribute to inflammation. And most of these are found within the body just normally. So first of all, we have histamine. This is going to be in response to injury, as well as mast cells in connective tissue, and basophils and platelets in the blood are all going to release histamine. Neutrophils and macrophages are going to be attracted to the site of injury and are also going to stimulate the release of histamine. This is going to cause that vasodilation and increased permeability of the blood vessels that we talked about before. Next, we have kinins. These are polypeptides that are formed in the blood from inactive precursors called kininogens, or these are the precursors. The substance is going to induce vasodilation, and it's also going to cause an increased permeability, and it's going to serve as a chemotactic agent for phagocytosis to help attract those phagocytic cells. Next, we have our prostaglandins. These are going to be really important these are going to be released by damaged cells and are going to intensify the effect of those histamine and the kinins. Prostaglandins are also going to stimulate the immigration of phagocytes through those capillary walls. Next, we have our leukotrienes. These are going to be produced by basophils and mast cells. And leukotrienes are going to cause increased permeability, and they're also going to function in adherence of macrophages to pathogens, and as well as functioning as a chemotactic agent that's going to be really useful in attracting those phagocytic cells again. And then last but not least, we have the complement. So there are different components of the complement system, and these are going to essentially stimulate histamine release. They're going to attract neutrophils via chemotaxis, and they're going to promote phagocytosis. And some components are also going to be involved in destroying bacteria. So we'll talk a little bit more about the complement system a little bit later, and you'll hear a lot more about it probably in microbiology class. So here we have a nice little table summarizing the different types of innate defense that we have, both internal and external. So now let's take a look at adaptive immunity. This is going to refer to the body's ability to defend itself against specific invading agents. So antigens are going to be substances that are recognized as foreign that are going to provoke some sort of immune response. And adaptive immunity is going to have both specificity and memory, and is therefore going to be divided into two types. We've got cell-mediated adaptive immunity and antibody-mediated adaptive immunity. So two properties that distinguish adaptive immunity from innate immunity are going to be, one, the specificity for particular foreign molecules, i.e. antigens, which is going to also involve distinguishing itself from non-self molecules. And the second property that distinguishes adaptive from innate immunity is memory for most of the previously encountered antigens, so that a second encounter is going to prompt an even more rapid and vigorous response. So let's take a look at how we produce these mature T cells and B cells. So like we mentioned before, adaptive immunity is going to involve lymphocytes called B and T cells. Both of these are going to develop in primary lymphatic organs, either in red bone marrow or in the thymus, and these are going to develop from pluripotent stem cells that are going to originate in the red bone marrow. B cells are going to complete their development in red bone marrow, and this process is going to continue throughout a person's life. T cells are going to develop from pre-T cells that are going to migrate from red bone marrow into the thymus, where they're going to finish maturing into fully formed, fully mature T cells. Most T cells are going to arise before puberty, but they are going to continue to mature and leave the thymus throughout life. So, before T cells leave the thymus, or before B cells leave the red bone marrow, they're going to develop immunocompetence, and this is the ability to carry out adaptive immune responses. So this basically means that B and T cells are going to begin to make several distinct proteins that are going to be inserted into their plasma membrane. So from these protein functions, some of these proteins are going to function as antigen receptors, as molecules that are capable of recognizing specific antigens. And there are two major types of mature T cells that are going to exit the thymus. We've got our helper T cells and our cytotoxic T cells. Our Our helper T cells are also known as CD4 cells, and these is basically going to mean that in addition to antigen receptors, they're going to have a plasma membrane, a protein on their plasma membranes. It's called CD4. So that's basically just how we recognize them, based on their additional cell surface markers. Our cytotoxic T cells are also referred to as CD8 T cells, and this is because their plasma membranes are going to not only contain those antigen receptors, but again, a protein that's known as CD8. And so this is where we get those names again. So looking at the different types of adaptive immunity, like we mentioned, we've got cell-mediated and antibody-mediated immunity. So cell-mediated immunity is going to refer to the destruction of antigens by T cells. And this is going to be excellent, very effective against intracellular pathogens, things like fungi, parasites, viruses. It's going to be really great against some types of cancer cells, and it's also going to be excellent for foreign tissue transplants. So cell-mediated immunity is always going to involve a cell attacking another cell. Then we have antibody-mediated immunity, also called humoral immunity, and this is going to refer to the destruction of an antigen by an antibody. This is going to work really well against antigens that are dissolved in body fluids, as well as extracellular pathogens, primarily bacteria, which are going to multiply in bodily fluids, but are rarely going to actually enter the body's cells. So often a pathogen is going to provoke both of these types of immune responses. So it's going to have both antibody-mediated and cell-mediated immunity are going to occur. So we need to talk just briefly about this process called clonal selection, just about the basic principle of it. This is going to be the process by which an immune cell is going to proliferate and differentiate in response to a specific antigen. So two major types of cells are going to result from this clonal selection. All right, we can either end up with an effector cell and a memory cell. All right, so effector cells are going to be the cells that are actually going to do the work of destroying that antigen. All right, so these cells are going to be cytotoxic T cells, our helper T cells, as well as a plasma cells, like a clone of a B cell. And then we have our memory cells. These are going to have a long lifespan, and they are going to provide a faster secondary invasion response to proliferating and differentiating into effector cells. So we end up with the production of both of these two cells. So we have one that's going to be stored for later so that we can recognize that invader if it comes again, and then we have the effector cell that's going to be responsible for dealing with and eliminating the foreign invader from the start. So those two are going to both be a result of this clonal selection. Both are very important. So now let's take a look at antigens and antigen receptors. So antigens are, just put simply, are chemical substances that are going to be recognized as foreign by an antigen receptor. And this happens when they're introduced into the body. Antigens are going to have two important characteristics. We've got immunogenicity and reactivity. Immunogenicity is the ability to provoke an immune response by stimulating the production of specific antibodies, as well as the proliferation of specific T cells. The term antigen is going to derive from its function as an antibody generator. Reactivity is the ability of the antigen to react specifically to the antibodies or cells that it provoked. So entire microbes, or just one part of a microbe, may act as an antigen. Typically, just certain little parts of a large antigen molecule are actually going to be the part that's going to trigger an immune response. And these small parts are called epitopes. So antigens are going to get past the innate defenses, the antigens that are able to get past innate defenses are generally going to follow one of three routes into the lymphatic tissue. Most antigens that enter the bloodstream, for example, are going to do so through, injured blood vessels, are going to be trapped as they flow through the spleen. Antigens could also penetrate the skin and enter the lymphatic vessels and lodge in the lymph nodes. And antigens that penetrate the mucous membranes are going to be entrapped by the mucosa-associated lymphatic tissue, that MALT. So those are our three main routes for an invading antigen to make it into the lymphatic system. Antigens are going to be large, complex molecules, and they're going to be typically going to be proteins, although sometimes they can be nucleoproteins or lipoproteins, glycoproteins, and on the odd occasion they can be these large polysaccharides. There are going to be specific portions of antigen molecules that are called antigenic determinants, also called epitopes, and these epitopes are generally what's going to be responsible for triggering this immune response. So the diversity of antigen receptors in both B and T cells is going to be the result of shuffling and rearranging a few hundred versions of several small gene segments in a process called genetic recombination that we get through mitotic cell division. Next, we have our majohistocompatibility complexes. So located on the plasma membrane of the body's cells are self-antigens. These are also called major histocompatibility complex antigens, or MHC antigens. These are transmembrane glycoproteins that are also called human leukocyte antigens, and this is because they were first identified on white blood cells. However, unless you have an identical twin, your MHC antigens are going to be unique to you. I should clarify. Unless you have an identical twin. If you have a fraternal twin, you're going to have unique major histocompatibility complexes. They're going to be unique if you're fraternal. So there are going to be several hundred thousand MHC molecule markers on the surface of each of your body cells, except for those red blood cells. Those have their own special surface markers that we mentioned before. So although our MHC antigens are the reason that tissues can be rejected when they're trying to transplant organs from one person to another, their normal function is to help T cells recognize an antigen that's foreign or not from your body. So that's their main role. So they are good to have. So we have several pathways for antigen processing. So for an immune response to occur, B cells and T cells must recognize that a foreign antigen is present. So B cells can recognize and bind to antigens in lymph, interstitial fluids, or in blood plasma. And T cells are only going to recognize fragments of antigenic proteins that are processed and presented in a certain way. So in antigen processing, antigenic proteins are going to be broken down into peptide fragments that are associated with their MHC molecules. And in a process called antigen presentation, our antigen MHC complex is then going to be inserted into the plasma membrane of a body's cell. So we have a couple of different pathways of antigen processing. So foreign antigens that are present in fluids outside the body are called exogenous antigens. And these are going to include intruders such as bacteria or any kind of bacterial toxins, those parasitic worms, any kind of inhaled pollen or dust that's going to give you those terrible allergies if you're, maybe living in the South, as well as any kind of viruses that have not yet infected the body's cells. A specific class of cells called antigen-presenting cells, or APCs, are going to process and present exogenous antigens. And those APCs are going to include our dendritic cells, our macrophages, and our B cells. So after processing and presenting an antigen, our APCs are going to migrate from tissues via the lymphatic vessels to the lymph nodes. So foreign antigens that are present inside the body's cells are going to be termed endogenous antigens. And these are going to be things like viral proteins that are produced after a virus infected the cell and takes over the cell's metabolic machinery. It can also be things like toxins that are produced from intracellular bacteria or an abnormal protein synthesized by a cancerous cell. So let's look at exogenous antigen processing. So the step one of exogenous antigen processing. This is ingestion of the antigen. This is when antigen-presenting cells are going to ingest exogenous antigens by phagocytosis or endocytosis. So ingestion could occur at almost anywhere in the body that the invaders, things like microbes and whatnot, have penetrated the innate defenses. Our second step is the digestion of the antigen into peptide fragments. So within the phagosome or within the endosome, protein digestion enzymes are going to split large antigens into short little peptide fragments. And then we have the synthesis of our MHC2 molecules. So at the same time, the antigen-presenting cells are going to synthesize our MHC2 molecules at the endoplasmic reticulum. And then we're going to have packaging of our MHC2 molecules. So once these are synthesized, our MHC2 molecules are going to be packaged into these little vesicles. From there, we have fusion of the vesicles, and this is when the vesicles that are containing the antigen peptides fragments and our MHC2 molecules are going to merge and fuse. The binding of the peptide fragments to the MHC2 molecules is our sixth step, and after this fusion of the two types of vesicles occurs, our antigen peptide fragments bind to our MHC molecules. Our last step is going to be the insertion of antigen MHC2 complexes into the plasma membrane. Right? The combined vesicle that contains the antigen MHC complexes is going to undergo exocytosis, and as a result, the antigen MHC complexes are going to be inserted into the plasma membrane of the cell. So that's how we do our exogenous antigen processing. Now taking a look at endogenous antigen processing, this is a little bit shorter. There are only 5 steps here. So we start with the digestion of antigens into peptide fragments, same as last time. So within the infected cell, protein digesting enzymes are going to split those endogenous antigens into short little peptide fragments. Then we have the synthesis of our MHC1 molecules. So at the same time as this digestion is occurring, the infected cells are going to synthesize MHC1 molecules at the endoplasmic reticulum. We then have binding of peptide fragments to MHC1 molecules. This is when the antigen peptide fragments enter the endoplasmic reticulum and bind to that MHC1 molecule. We then have packaging of our antigen MHC1 molecule complex. This is so from that endoplasmic reticulum, our antigen MHC1 molecules are going to be packaged into vesicles, and then we're going to have the insertion of antigen MHC1 complexes into the plasma membrane. So the vesicles that contain MHC1 antigen complexes are going to undergo exocytosis, and as a result, the antigen MHC complexes are going to be inserted into the plasma membrane. Last but not least, a little bit that we have to cover are cytokines. We're really not going to talk too much about these. I've got a nice little table here if you want to look at the cytokines that are involved in the immune response, but don't worry too much about it. Just simply know that cytokines are small protein hormones that are going to stimulate or inhibit many normal cell functions, things like cell growth and differentiation. However, lymphocytes and antigen-presenting cells are also going to secrete cytokines, as do fibroblasts, endothelial cells, monocytes, hepatocytes, and kidney cells. So lots of things are going to secrete these cytokines, and they're going to have plenty of normal functions in the body. However, some of them, which are listed, some of these cytokines, which are listed in this table, are going to play a role in causing an immune response in the body. So just basically just know that. Don't need to get too in depth with any of these. So that is all that we have for Chapter 22 on the lymphatic system. As always, if you have any questions, comments, or concerns, please feel free to contact me or ask after class. So thanks so much and have a great day.